So, hello students, welcome back to CBSE and NCERT Solutions. This is Basraj sir, your biology master teacher. Now, in this channel, we make NCERT line by line sessions. We conduct NCERT line by line sessions, and also we do something called as your complete backside NCERT solutions. Right, whatever is questions are given in your backside of your every chapter, we also do that also. So, students, let's start with the second part now. So, all of you remember biomolecules, the first part which we did, right? In the biomolecules first part, we finished something called as lipids. We finished something called as your proteins to some extent, amino acids. I thought you. Then we also did other basic structures. For example, we did something called as retentate and what is filtrate. So all that was done in the first part. So what are we going to do in the second part now? Right? What are we going to do in the second part? So in the second part is very important. Let me tell you first of all because many of the questions, right? One second, student. Let me just check if I'm audible. Yeah, I'm audible perfectly. So many of the structure and many of the questions usually come from the second part. And the second part is a part where many students struggle with the concept of Km value, Vmax value, and what is the meaning of enzyme, and what is this cofactors, what is this competitive inhibitors, uncompetitive, non-competitive inhibitors. So all of this difficult part of this chapter biomolecules we'll be dealing with in the second part right now. Okay, so with that being said, let's start today's class by starting off with polysaccharides. Now, all of you remember the basics of polysaccharide, right? All of you remember polysaccharide, right? Polysaccharides, obviously, their molecular weight, the weight of the polysaccharide is more than 1000 daltons. It is more than 1000 daltons. If the molecular weight is more than 1000 daltons, now you tell me in the chat, all of you, do you find polysaccharides in the retentate or filtrate? You will tell me, obviously, sir, polysaccharide is a macromolecule and its weight is since more than 1000 daltons. It will be found in the retentate on the top. On the top. Now, all of you remember also every single molecule which is found in the retentate on the top is also called as what? Acid insoluble. Yes, it is also called as what? Acid insoluble fraction. Now, those of you who are not able to follow this, try watching the first lecture where we have discussed about the difference between acid soluble and acid insoluble fraction. Okay. Now, acid insoluble pellet also has polysaccharide, that is mostly made up of carbohydrates as another class of macromolecule. So carbohydrate is also a macromolecule. Now you can also argue here, students, sir, in polysaccharides, don't we have something called as, in carbohydrates, don't we have something called as monosaccharides, disaccharides, oligosaccharides? Remember, students, most of your monosaccharide and disaccharide, they will be somehow found in the filtrate. But since poly means what? Poly means many. Poly means many. So if many monomeric units are joined, obviously molecular weight is higher, and that is why polysaccharides are found in the retentate on top. Okay. Polysaccharides are long chain of sugars. I told you the long chain of sugars, long chain of monosaccharides are joined together, glycosidic bond, and they are found in the monosaccharide form. Their thread, literally a cotton thread. That means how fine they are, right? Containing different monosaccharides as building blocks. For example, cellulose. Cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide. Now, friends, let's get into it one by one. For example, let me teach you what is a carbohydrate first of all. Before we go any further, carbohydrate. Carbohydrate is nothing but they are basically the hydrates. They are the once again they are the hydrates of carbon. They are hydrates of carbon. And if you look at the structure of a carbohydrate, do you notice they have something called as multiple OH groups, hydroxyl groups? Can I say they have something called as multiple OH groups? They have something called as multiple OH groups. At the same time, if you look at a carbohydrate, do you see they have something called as, in this case of glucose, they have an aldehyde group. They have a aldehyde group. And at the same time, if you look at a fructose, structure of fructose, it has a ketone group. It has a keto group. Keto group. One second, students. It has a keto group. Right. Now, let's learn the basic classification of your carbohydrate. Basic classification, the most basic. For example, there is something called as monosaccharide. Monosaccharide. Monosaccharides. Now, what is the meaning of mono? Mono means one. So it is made up of only one sugar. It is made up of one sugar. One sugar molecule. One sugar molecule, that's all. And since it is made up of one sugar, you cannot hydrolyze it even further. So meaning of hydrolysis is what? You take a complex sugar, you hydrolyze it and you get a simple sugar from it. But monosaccharide is always already the simple sugar. Monosaccharide is already the simplest. It's already the simplest carbohydrate. It is already the simplest carbohydrate. You cannot hydrolyze it further. It means you cannot make it more simple than what it is already. So that is monosaccharide. Example, all of you know. Glucose is the best example. Glucose is a crazy easy example to remember for monosaccharide. For monosaccharide. Now, can anyone tell me in the chat, what about disaccharide? What about chat? Anyone known is responding in the chat today? What about disaccharides? What are they made up of, students? Can anyone tell me in the chat? What are disaccharides made up of? No one is answering in the chat today. Students, I hope I'm visible now. I feel my half of the face was already gone. I moved little. So I hope I'm visible properly now. Now disaccharide, students. Disaccharide is made up of two sugar molecules. It's made up of two sugar molecules. Made up of two sugar molecules. That is a disaccharide, right? That is a disaccharide. Made of two sugar molecules. That's a disaccharide. Now, students, I'll give you a quick example. Let's see how many of you can understand. I'll give you a quick example. Okay. For example, sucrose. Sucrose. One second, students. I'm trying to adjust myself, but it's not getting adjusted. I don't know why. I think I moved a little somewhere. I moved. Okay. Can anyone tell me what is sucrose made up of? What is sucrose made of? Anyone in the chat who can tell me what is sucrose made up of? Anyone? What is sucrose made up of? Can anyone tell me in the chat what is sucrose made up of? All of you know sucrose is made up of glucose plus fructose. Glucose plus fructose can give you the sucrose. Now you will ask me, sir, how are they attaching? Like, how can I tell glucose is forming a bond with fructose? How are they attaching? Are we adding some glue there? No, student, there is no particular glue here, but there is a bond formation here between them. There is a bond here though. The bond is called as your, anyone who is answering today? Okay. The bond is called as glycosidic bond. Glycosidic bond. Glycosidic bond is present between the two monosaccharides. Between the two monosaccharides, glycosidic bond, right? Glycosidic bond is present. Now, students, similarly, there is something called as oligosaccharide also. Similarly, there is something called as oligosaccharide. You need to understand that oligosaccharide is made up of more than more than two monosaccharides. So when more than two monosaccharides join together, they make up the oligosaccharide. Example is raffinose. Raffinose is a good example. But what are we interested in? We are interested in the polysaccharide friends. What we are interested in is the polysaccharide. Our main interest is in the polysaccharide. Okay, main interest is polysaccharide. Understand? Polysaccharide is made up of, it is basically a polymer. It is basically a polymer of monosaccharide. It is a polymer of monosaccharide with 10 or more repeating units. 10 or more repeating units. When 10 or more repeating units come together, they make up the polysaccharide. Now, polysaccharides. Now, students, I'll give you a small classification here. Small classification for polysaccharides. Okay, understand? Very simple. Very simple. Understand. For your monosaccharides can be broadly classified, students. Monosaccharides can be broadly broadly classified into something called as homopolysaccharide and heteropolysaccharide. Understand the difference. Homopolysaccharide saccharides as well as heteropolysaccharide. Homo and heteropolysaccharide. What is the difference? Now I told you, polysaccharide is basically multiple repeating units. In your school also does it happen? We have many students, but in one day when there is color dress or you wear the uniform color house uniform, all the house is standing one side, all the green house is standing one side, and all the yellow house and blue house standing one separately. All of you are studying together based on the uniformity, yes, based on the house. Similarly, in the case of in the case of homopolysaccharides, all monomers, all monomers are same sugar. All repeating monomers are same sugar. Glucose, 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 glucose will be long chain of just glucose. That is called as homopolysaccharide. Then what is heteropolysaccharide, friends? Heteropolysaccharide is basically different type of sugar. Example, I'll tell you. NAG and NAM. NAG and NAM. Repeating units of NAG and NAM is called as heteropolysaccharide. Okay, so I hope that is clear to all of you. What is the difference between homopolysaccharide and heteropolysaccharide? In the case of homo, means same monomers there, repeating units of same monomer. Okay, now friends, now I will teach you something called as classification of homopolysaccharide. Homopolysaccharide. What are the different types of sugar which we have? Homo and heteropolysaccharide. This is the broad classification, right? This is the broad classification. This is just the broad classification. Broad classification. Now, under homopolysaccharide, we have two categories. Under homopolysaccharide, we have two main categories. The first one is for structure. Structural. The second one is for storage. It's for storage. Under structural, we have something called as cellulose. Cellulose. And we also have something called as chitin, which make up the external layer. Cellulose in the case of plants, chitin in the case of insects. Under storage, we have something called as starch. In the case of plants, which you find, right? Starch. We have glycogen, which is the storage material in the case of animals. Then we have something called as inulin. We have something called as inulin. Inulin is also a food stored material in the case of your plants. In the case of your plants, inulin can also be found in the case of your plants. Now, what about heteropolysaccharide, friends? Heteropolysaccharide mainly has two different examples. One all of you already know, that is peptidoglycan. And peptidoglycan, we have something called as, it is made up of NAG and NAM, peptidoglycan. The other heteropolysaccharide is agar-agar. It is the agar-agar. Agar-agar is also a heteropolysaccharide. It is made up of different types of repeating unit. Two types of repeating units. Okay. Now, see this, all of you. I hope all of you are able to see this, right? I hope all of you are able to see this now. I hope all of you are able to see this. Under storage, what do we learn? Under storage, we learned about starch, glycogen, inulin, not insulin, inulin. Under structural, we learned about cellulose and chitin. Now understand here for starch, it is first of all polymer of glucose, meaning is that starch is made up of repeating glucose units. The entire starch is made up of glucose, 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 glucose. Alpha 1 for linkage. In the case of plant, reserved food material in the case of plants consist of amylopectin and amylose. You will learn this more in chemistry. The structure you will learn in chemistry. Now what about glycogen? Glycogen also is a polymer of glucose, repeating units of glucose. Then what is the difference between your starch and glycogen then? The difference between starch and glycogen is that the branching. For example, in the case of glycogen, the branching is much more higher. That is, alpha 1,6 linkage is much more better here in the case of glycogen. Okay, highly branched, stored in liver and muscles. Stored in liver and muscles. Okay. Now, what about inulin? Inulin is a polymer of fructose. Very important. Also, previous year question for NEET examination, PYQ, NEET PYQ. This is the inulin is a polymer of, like for example, how we learned that glycogen. In glycogen, we have glucose, 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 glucose. In the case of inulin, we have fructose, 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 fructose. Long chain of fructose. That is in the case of inulin. In the case of inulin, long chain of fructose. Not metabolized in human body, stored in the helia, dandelions, and aristochoke. That is mostly found in the case of plants. Annually. Now structure, all of you know, again it is a polymer of glucose. Polymer of glucose, cellulose, glucose, 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 long chain of glucose will be there. And all of you know it is found in the case of your plants, cellulose, outer layer of the plants. Okay, outer layer of plants. Okay, straight chain unbranched, straight chain unbranched. Now chitin, polymer of N-acetyl glucosamine, NAG. So NAG, long chain of NAG will be there. That is what is made up of chitin. Chitin is made up of long chain of NAG, NAG molecules. Okay. And where do you find it? You find it in the arthropods. Exoskeleton of arthropod, cockroach basically. In your house, if you look around, you'll see some cockroach. In that cockroach, outer exoskeleton is there, right? On the exoskeleton, it is made up of chitin. Okay. Good afternoon, MD Munna Bhai. Okay. It is a complex polysaccharide. We will get to this. We will get to this. Now, students, apart from that, we have heteropolysaccharide also. Hetero means what? Hetero means different. So, different types of polysaccharide. I told you already in the case of peptidoglycan, it is made up of NAG and NAM. NAG and NAM. You can see here. N-acetylglucosamine, N-acetylmuramic acid, long chain of NAM, and there will also be NAG here. NAG and NAM will be your repeating units. Peptidoglycan. Two types of monosaccharides repeated continuously. Okay. Now look at agar agar. Agar agar is also made up of galactose and 3,6 anhydro L galactopyranose. Okay. That these two are different types of sugar. These different types of sugars are repeating units. Repeating units. Okay. I hope the concept is clear. That is the difference between homopolysaccharide as well as heteropolysaccharides. Okay. Homopolysaccharide and heteropolysaccharide. Clear? Now we will understand the next concept. It is called as your reducing sugar. Reducing sugar you might have heard. Like you might have heard about reducing sugar when we learn about your in chemistry. Reducing sugar, non-reducing sugar. So what is the meaning of reducing sugar, students? Meaning is very simple. The meaning is very simple. Reducing sugars are a type of carbohydrate. Right? They are a type of carbohydrate. They are a type of sugar which has a free aldehyde or a ketone group. If you look at the glucose, it has a free aldehyde. Look at fructose, a free ketone group. If you look at galactose, a free aldehyde group again here, aldehyde group. So if there is a free aldehyde or a ketone group, it is present, then it is called as a reducing sugar. Now you will ask me, sir, what is so great about this, presence of free aldehyde or ketone? The greatness is very simple. This free aldehyde or ketone, right? This free aldehyde or ketone can react with some other chemicals. For example, here you need to take 2 ml of glucose. 2 ml of glucose. If you're taking 2 ml of glucose, you need to add 2 ml of Benedict solution. 2 ml of Benedict solution. Equal amount. Now keep it in water bath. Keep it in water bath. Then you will see something. Then you will see what happened. The blue color is turning into brick red. Now, what is the reaction, students? The reaction is very simple. What has happened is the reducing sugar reduce the cupric ions to cuprous ions. This free aldehyde or ketone groups, this free aldehyde or ketone groups are reacting with the cupric ions. Cupric ions are present where? Cupric ions are present in the Benedict solution. Okay, so they're taking aldehyde ketone is reacting with cupric ions and converting the Benedict solution from the blue color to red color because cupric is turning into cuprous. Now, what about your These all three are monosaccharides, right? They are monosaccharides. Now, what about disaccharides, students? What about disaccharides? The disaccharides as well as your polysaccharides are non-reducing sugars. They are non-reducing sugars. Now, you might ask me, sir, why are they non-reducing? Why do you call them non-reducing? Because, students, answer is very simple. Because non-reducing sugar do not have any free aldehyde ketone group. Aldehyde ketone group is completely absent here. A free aldehyde or a free ketone group is absent in the case of your non-reducing sugar. That is in the case of your polysaccharides and disaccharides. No free aldehyde ketone group. Now, I'll give one more test, students. Ready for one more test? The one more test is also very simple. It's called as iodine starch test. So what you need to do is you need to take iodine solution to a starch containing sample. So we have starch. This is the starch. Okay. So this iodine is added to the starch here. It becomes blue color. Iodine becomes blue color. At the same time, I am taking in this particular solution, I'm taking cellulose. Taking cellulose. If you take this iodine and add it into cellulose, then it will color. There will be no prominent coloration. The color will almost remain the same. Why? The concept is very simple. For example, students, if I'm throwing this pen at you and I will tie your hands, will you be able to catch it with your hand? Someone will say I will catch it with my mouth and all, but that is lies. But will you be able to catch with your hand? You won't be able to catch with your hand. It will be very difficult to catch with your hand. At the same time, if I don't tie your hands and I throw the pen at you, that time can you catch it? Can you catch it? Yes. Some of you will still drop it, but it's a different story. But will you be able to catch it? Yes, you'll be able to catch it because your hands are free, your fingers are free. So look at the structure of your starch and look at the structure of cellulose. If you look at the structure of a starch, does it have free hands? It has branching. Yes, it has branching, and branching will show that it has free hands. That is, it can catch iodine. But at the same time, if you look at the structure of cellulose, if you look at the structure of cellulose, it does not have any type of free hands. All the hands are legit tied here. Every single hand is tied nicely, and they're like, I will not leave your hand. So what we're trying to understand here is, since the structure of starch is different from structure of cellulose, and if you look at the structure of starch, it has something called as amylopectin, which has alpha 1,6 linkage, and it has branching, and all the iodine can be caught in that branches. It can be held in the branches. But at the same time, if you look at the cellulose, there is no branching, and that is why iodine cannot be held in the cellulose. Okay, so you cannot hold iodine in cellulose, but you can hold iodine in the starch because of branching. That's all. See this question, students. See this. Proteins. Proteins are polypeptides. Meaning of poly means many. Peptide is basically the amino chain up to 20 amino acids. Polypeptide. They are linear chains of amino acid linked by peptide bond as shown in the figure 9.3. We will see the figure later on, but to understand what is the polypeptide, polypeptide is basically amino acids are joined together by peptide bond. Long chain of amino acids. Long chain of amino acids. Okay. Now, each protein is a polymer of amino acid. Obviously, protein is made up of what? Protein is made up of amino acids. As there are 20 amino acids, for example, out of 20, how much you know, right? Out of this 20 amino acids, 15 are neutral, 3 is basic, and 2 is acidic. 2 is acidic in nature. That is the general rule of amino acids. 20 amino acids. A protein is a heteropolymer and not a homopolymer. Meaning is what? Meaning of heteropolymer is that it is made up of different types of monomers. Obviously, each amino acid is a polymer, each amino acid is a monomer. So many amino acids polymer. And it is homo. No, it is heteropolymer. Why? Because each polymer is different. Because all of you know there are 20 different amino acids. All 20 different amino acids can form a long chain. Yes, they can form a long chain. Right? A homopolymer has only one type of monomer repeating units n number of times. That is in the case of homopolymer. Homopolymer will have only, for example, it will only have lysine. It will have only lysine. Continuously only lysine. Continuously only and only lysine. Now this becomes a homopolymer. Now what a heteropolymer? Heteropolymer will have lysine first. Then it has something called as glutamic acid. Glutamic acid. Then you can also have your aspartic acid. Then you can also have arginine. So basically the entire amino acids can differ. When the amino acid chain is different, this is called as heteropolymer. Heteropolymer. Okay, it's called as heteropolymer. Now, this information about the amino acid content is important as later in your nutrition lessons you will learn about certain amino acids are essential for our diet. For example, there are certain amino acids which we take through our diet because they are not produced in our body. If something is not produced in your body, you will take it from outside. For example, if they don't make chicken biryani in your house, but you want to eat chicken biryani, what do you do? You eat from the outside. Same thing. If some amino acid is not prepared in your body, you take it from the outside. And that becomes the essential amino acid. Why is it essential? Because it is not produced in your body. It is not produced in your body. That is why it is called as essential amino acid. Okay. Now, what about if some amino acid is prepared in the house? Will you buy it from outside? For example, your mother is making anna sambar every single day. Very rarely you will go buy anna sambar from outside because it's already made in the house. Similarly, if some amino acid is made inside our body, it is called as non-essential. It is not very important because it is already produced a lot. Okay. Hence, dietary proteins are a source of essential amino acid. Dietary source means the source which you are eating from outside. The outside source are good and non-essential amino acid. Therefore, amino acids can be essential or non-essential. The later are those which our body can make, non-essential or body can make. While we get essential amino acids through our diet and food, proteins carry out many functions in living organisms, some important nutrients across the membrane. Some fight infection, and some are hormones and some are enzymes. Basically, the different functions of the enzymes is given here. Different functions of protein. Some protein can become intestinal ground substance, collagen. Some examples of proteins are enzymes, right? Trypsin and all. Some of the proteins are hormones, insulin. Some are antibiotics, some proteins. Some proteins are receptors in receptor nature. Some proteins will help in transportation. For example, here I have one cell. This is basically glucose. Glucose is not able to enter the cell. What will it do? Glucose will form a partner that is called as GLUT4. GLUT4. Now, GLUT4 will help the entire complex to enter inside because that can enter inside very easily. Okay. The GLUT4 enables oxygen transport into the cell. Oxygen is entering inside the cell. Okay. Collagen is the most abundant protein in the animal world. All of you know collagen is the most abundant protein in the animal world. But what about the whole biosphere? When we take the entire biosphere, that time it is the Rubisco. Why? Because Rubisco is found in the case of your plants. Both C3 and C4 plants. So obviously all of you know plants are higher in number. So all the organisms will have plants will have C3 and C4. Yes, they will have C3 and C4. Right. So C3 and so your Rubisco is the most abundant protein in the world. We did this already, right? Cellulose is a polymeric polysaccharide consisting of one or two monosaccharides. Cellulose is a homopolysaccharide, we told that. Starch is a variant of this, but present as storage. All of you remember here, we learned it here. So here, cellulose and chitin is storage. Starch, glycogen, inulin is sorry, starch, glycogen is storage. Cellulose and chitin is structural. Okay. Animals have another variant called as glycogen in the case of animals with high branching. Inulin is a polymer of fructose, PYQ. It's a glycogen. The right end is called as reducing end. The left end is called as non-reducing end. All of you. Right is reducing. R for right. R for right. R for reducing. Reducing end. Which has a free aldehyde or ketone. Okay. It has branches shown in the figure. Okay. Now, very important line again. Starch forms helical secondary structures. In fact, starch can hold I2 molecule. I told you, starch can hold I2 molecule. Why? Because it has the fingers, it has the branching in the helical portion. The I2 is blue in color. Cellulose does not contain complex helical and hence cannot hold I2. That is, for cellulose, I have tied the hands. If I'm tying the hands of cellulose, will it be able to catch the pen? No. Will it catch the chemicals? Will it catch the color? Will it catch the iodine? No, it will not catch the iodine also. Okay. Now here they're showing you the complex one for linkage. Alpha one for linkage. This is 1 6. This one is 1 6. This one. Plant cell pulp are made up of cellulose, all of you know that. Paper made up of plant pulp, called as cellulosic in nature. The N-acetyl glucosamine, N-acetyl glycosamine, and N-acetyl glycosamine are found in the exoskeleton of arthropods. That is on the cockroach. Top of the cockroach they can be found. Okay. Chitin. These complex polysaccharides are mostly homopolymers. So write down one concept here called as complex polysaccharides. The first one is your chitin. Then we have glucosamine. Then we have the N-acetyl galactosamine. Complex polysaccharides. Three are examples of complex polysaccharides. Now, students, no. We'll take a 15 minutes break and we'll come back and we'll do nucleic acids that time. 250, right? Okay. 3 5 p.m. Resume the class. Yo. Now we will do something called as nucleic acids, right? Again, very easy, students, to solve nucleic acids. Now, nucleic acid is also a type of macromolecule. It is also a type of macromolecule. Now tell me, if it is a type of macromolecule, if it is a type of macromolecule, where do you find it? Can anyone tell me in the chat? If it is a macromolecule, do you find it in the retentate or filtrate? Obviously, you'll find it in the retentate. Right, retentate. Why? Because the molecular weight is more than 1000 daltons. More than 1000 daltons? Yes. Okay. Nucleic acids. Nucleic acid that one would find in the acid insoluble pool. So if it is in the retentate, obviously acid insoluble pool, okay, of any living tissue in nucleic acid is the nucleic acid. There are polynucleotides. What does the meaning of poly? Poly means many, nucleotide. Nucleotide, I've told you in previous class, it has a sugar group, a phosphate group, and a nitrogenous base. That is a sugar group, it has a sugar, it has a nitrogenous base, and it has a phosphate attached to it. That is a nucleotide. When many nucleotides join together, that makes up the nucleic acid. Okay. Together with polysaccharide and polypeptide, these are comprising the true macromolecules. So polysaccharides, polypeptides, and polynucleotides. I'll write them for all of you. Otherwise, you'll forget off. I know for sure. Polysaccharides, polysaccharides, polypeptide, as well as polynucleotide, polynucleotides. All three of them together make up the biomacromolecules. They are the macromolecules, biomacromolecules, all three of them. Okay, biomacromolecules. Now, for nucleic acids, the building block is nucleotide, I told you. So nucleic acid is what? Made up of nucleotide. A nucleotide has three chemically distinct components. One heterocyclic compound, the second is monosaccharide, and third one is phosphate. So one is nitrogenous base, sugar, and phosphate. Three makes up the nucleotide. Now, the heterocyclic compound is the nucleic acid are the nitrogenous bases. So, heterocyclic is a nitrogenous base. It could be adenine or it could be purine or any one of them. It could be like purines and pyrimidines, like adenine, guanine, aspirate purines, while pyrimidines are your uh, while the rest are the pyrimidines. The skeletal heterocyclic ring is called as purine and pyrimidine respectively. The sugar can be of two types again. A sugar can be of deoxyribose or the sugar can be ribose. Now you might wonder, sir, where do you find deoxyribose and where do you find ribose sugar? Your deoxyribose sugar, friends, your deoxyribose sugar is found in the case of DNA. Deoxyribose. What is the meaning of that? One oxygen is missing. In the case of ribose is found in the case of RNA. Now, if you look at the structure of deoxyribose, in the second carbon, can you see one oxygen is missing here? While oxygen is present in the case of ribose. So, deoxy, missing of oxygen is happening in the case of deoxyribose. Now, this any one of those sugars can be found in the nucleotide. Found in the nucleotide. Okay. All of you know purines, we have guanine and adenine. Then you have pyramids. This is a small trick to remember pyrimidines. What is the trick to remember pyrimidines? Cut the pyramid. Cut the pyramid. Pyrimidines, cytosine. This is your cytosine, uracil, thymine. Cytosine, uracil, thymine. Cut the pyramid. Okay. Remaining two are your purines. Now see here. Let's see what is given here. The sugar is found in the, is either a monosaccharide is either ribose or deoxyribose. The nucleic acids containing deoxyribose is called as DNA. DNA has the deoxyribose sugar. RNA has the ribose sugar. RNA has the ribose sugar. Now, students, now we will understand proteins. The structure of protein, what is the meaning of primary structure, secondary structure, tertiary structure. All of them we will learn right now. All of that we will learn right now. Okay. Let me finish this. Yeah. Now, what is the primary structure of a protein? Understand the concept, students. Understand the concept. In the case of primary structure of protein, we have amino acids arranged in a linear fashion. You see, it is arranged in a linear fashion. Then what do you understand from the primary structure? What is the significance of primary structure? The significance of primary structure is that you can tell which amino acid is present where. For example, if I make all of you stand in one single line with one arm distance, can I tell exactly who is standing where? Can I tell who is standing where exactly? That information is given by the primary structure. I can tell we have prime amino acid 1, 2, 3, 4, 5, 6 and so on. You can exactly tell which amino acid is present where from the primary structure. Now what about secondary structure, students? Secondary structure. Secondary structure can be of two types. For example, first one is called as alpha helix, other one is called as beta plate sheet. This is a much more complex version or complex arrangement of primary structure. So take the primary structure and you add hydrogen bonding. You need to add hydrogen bond. Hydrogen bond. Here we only had peptide. In primary we only had peptide. Okay. Now this alpha helix will be like a solenoid. In the physics you might have learned no the solenoid model. Now what is tertiary structure? Very simple again. You take the alpha helix, you take the beta plated and nicely make a woolen ball. You take the alpha helix, you take the beta plated and nicely make a woolen ball like structure. That is called as tertiary structure, and tertiary structure is always 3D. It is always three-dimensional in nature. And most of the biologically active enzymes, the most of the biological active enzymes are at the tertiary level. They are at the tertiary level. Now, what is quaternary structure then? What is quaternary structure? Very simple again. You take one tertiary from here, take one tertiary from here, and join the two tertiary. When you join the multiple tertiary structures, we end up getting the quaternary structure. For example, if you look at this diagram right here, this is a one chain, separate chain. This is second chain. Two chains are joined together in the case of tertiary structure. I'll give you a small revision again. Understand. Primary structure is linear. I told you, linear chain of amino acid. Present represents the position of amino acid. It tells you which amino acid is present where. Here it has something called as N terminal. Here, N terminal is the first terminal. N terminal is the first terminal. Why do we call N terminal as N? Because it has a free amino group. Free amino group. Okay. Now, last terminal is called a C terminal. Why? Because it has a free carboxylic acid group. C for carboxylic acid, C for C terminal. Okay. So N terminal is the first, C terminal is the last. No. Second structure. The first one is called as alpha helix. Can you see? Alpha helix, which will have hydrogen bond like this. It will have hydrogen bonds here. Hydrogen bonds here. Alpha helix. Then we also have something called as beta plated. This is called as beta plated structures. Okay. Now tertiary structure. This is where they'll ask you a question also, students. I told you first of all it's the 3D structure. Protein fold upon itself. You take a alpha helix, take a beta plated, and nicely make a woolen ball out of it. That gives you tertiary, and it is biologically active in nature. Okay. Now, quaternary. Quaternary, what is happening? We have one peptide chain on top, one peptide chain on the bottom. When many tertiary are coming together, we end up getting the quaternary. Two or more polypeptide chains are here. Example, I'll tell you best example is hemoglobin. Right, hemoglobin, human hemoglobin is made up of four chains. See, chain 1, chain 2, chain 3, chain 4. There are two beta chains and there are two alpha chains. Two alpha and two beta chains are here. Okay, in the case of quaternary structure, hemoglobin is example. Let's read what is given in NCERT. Let's read what is given in your NCERT also at the same time. A protein, as mentioned earlier, are heteropolymers. Right? Proteins I mentioned earlier, they are what? Heteropolymers. Containing strings of amino acid. The structure of a molecule means different in many different languages. Let's understand what is there in the case of biology. See, the sequence of amino acid, that is the positional information in a protein. That is, which amino acid is present where, which is the first amino acid, which is the second amino acid. All that information is given by the primary structure of protein. A primary structure of protein will tell you which amino acid is present where, basically. Okay. Now, a protein is imagined as a straight line. The left end represents the first amino acid, also called as the N terminal. N terminal. Okay. The first amino acid is called as N terminal. Last is called as C terminal. Why C terminal? Because of the carboxyl group. Because of the carboxyl group. Okay. Now, a protein thread does not exist throughout as a fixed and rigid rod. The thread is folded in a form of helix. I'm telling you, a thread can be folded in the form of helix. Solenoid model, that is called as alpha helix. Alpha structure. Other one is beta plated structure. Okay. In a protein, only right-handed helix are observed. The other regions of the protein's thread are folded upon secondary structure. No. What is secondary structure? The long protein chain is also folded upon itself. So if you take alpha, if you take beta, and you fold it nicely and make woolen ball, that will give you the tertiary structure. Tertiary structure. This gives us a 3D dimensional view of a protein. Tertiary structure is absolutely necessary for many biologically active proteins. Without tertiary structure, proteins cannot be active. Quaternary structure, all of you know. Some proteins are assembled of assembly of more than one polypeptide. So if you take one polypeptide chain, take one more polypeptide chain and join the two polypeptide chain itself. When we join multiple polypeptide chain, we end up getting the quaternary structure. Quaternary structure. Okay. Adult hemoglobin consists of four subunits, 2 alpha, 2 beta. So four different chains are coming together and they're forming the quaternary structure. Okay. Difference. Again, now I'll tell you something called the nature of bond. Which type of bond is present basically? Peptide bond is present where? Peptide bond is present between two amino acids. It's formed between a carboxylic acid and amino group of an amino acid. All of you remember a structure of amino acid? NH3, NH2, and this is R. This is going to be H. Simple amino acid. Okay. Hmm. COH group of, that is a peptide bond between the amino acids. Glycosidic bond is found where? Glycosidic bond is formed between the two carbohydrates. Now students, till now what we learn? Till now we learned that a protein can have structure, it can function in structure, contractile, nutrition, regulatory, defense mechanism, that is antibiotics, transport, GLUT4. But a particular protein can also have something called as catalytic activity. It can have something called as catalytic activity. It can help in some kind of reactions. It can help in some form of reactions, that is called as catalytic activity of a protein. For example, friends, what is a catalyst? What is a catalyst? Before you understand catalyst, I want everyone to understand the definition here. What is the definition of a catalyst, friends? A substance, a substance which alters the rate of reaction. A substance which alters the rate of reaction. It can increase the rate of reaction or decrease the rate of reaction. Right? It is not always it increases rate of reaction. It can increase the rate of reaction or decrease the rate of reaction. But always remember, students, always remember it is not consumed in the reaction. It is never consumed in the reaction. Never. A catalyst will never be consumed in the reaction. Okay? It will never be consumed in a reaction. Now, catalysts can be of two types, mainly, broadly speaking, two types. We have organic catalyst and inorganic catalyst. Organic as well as inorganic. Under inorganic, which you use in chemistry, platinum, all that. Okay. Under organic catalysts are called as your enzymes. They're called as what? Enzymes. Organic catalysts are called as your enzymes. Okay, enzymes. Example trypsin. Example trypsin. Trypsin is a good example of enzyme. Okay, that is a catalyst. Let's understand about enzymes now. Let's understand about enzymes. What are enzymes made up of, and where do you find them and everything, and what is the best requirement of conditions for enzyme? Okay, what is an enzyme, students? What is an enzyme first of all? All enzymes are basically nothing but proteins. All enzymes are proteins. All enzymes are proteins except ribozyme. Except the ribozyme. Ribozyme is a RNA enzyme. It is an RNA enzyme. Ribozyme is an RNA enzyme. Okay. Now, enzymes are very highly specific. They are highly specific in nature. Highly specific in nature. They'll make sure they always bind with a very specific substrate. They'll not go here and there. They're very specific. This is my substrate. This is the enzyme. Enzyme substrate complex. Like one key, one lock. One key, one lock. Okay, always. Always one key, one lock. Always one key, one lock. Okay, that is highly specific in nature. That is enzymes are very highly specific in nature. Okay, now understand. So I'll teach you now something called as active site. Very simple concept. Understand what is the meaning of active site right now. For example, this is the structure of a protein. Let's say I'm taking quaternary protein. This is an enzyme or a quaternary structure of a protein. All of you know quaternary structure are biologically active. Now, in this structure of an enzyme, on the structure of an enzyme, there are several gaps or pockets. Can you see here? In this enzyme right here, there are small gaps or pockets are also called as crevices. If you can see here, there are small gaps or pockets, or I can call it crevices. Crevices on the structure of the enzyme. On the structure of the enzyme, these small gaps or the pockets or crevices, students. Small gaps are called as, they will have something called as a functional group there. They will have an R group. These particular structures are called as active site. Active site. So active site is what? Active site is a crevice or a pocket in the structure of, or in their structure, or structure of what? Structure of enzyme for binding to the substrate. Very specific in action, catalyze the reaction at high rate. That is an active site. And remember, students, always this concept you should remember it. An enzyme will never go and bind to the substrate. In fact, a substrate will come and bind to the enzyme. I'll repeat again. Our enzyme will never go behind the substrate. It's like combined with me, combined with me. Never goes. Enzyme is high demand. All the substrates will come and join with the enzyme. It's always ulta. Okay, substrates are very free. They are always free. They don't have too much time on their hand. Every single time substrate will see an active site, it will run behind the active site and join there. Okay. Now enzymes. Almost all enzymes are proteins. I told you. Almost all enzymes are proteins. Almost all enzymes are made up of protein except the ribozyme. Except the ribozyme. Ribozyme. One can depict an enzyme by a line diagram. An enzyme can be any protein that has a primary structure, that is amino acid sequence of a protein. An enzyme, like any protein, has a secondary and tertiary structure also. So an enzyme can have primary, secondary, tertiary structure. Why? Because enzymes are basically proteins. Enzymes are basically proteins, right? Now, when you look at the tertiary structure, you will notice the backbone protein chain fold. Backbone of the protein chain folds. See here, upon itself. The chain crisscross itself, and hence many crevices or pockets are made. So on the structure of the tertiary structure of protein, on the structure of the tertiary structure of protein or the enzyme, you will have small gappets, small gaps and pockets on the top of the enzyme. And the small packets or pockets or crevices is called as active site. It is called as active site. An active site of an enzyme is a crevice or pocket into which substrate is fitting. So substrate will come and it will join on the active site. Okay. Now, thus enzyme through their active side catalyze the reaction at high rate. So they can catalyze the reaction at a higher rate because of the enzyme. The enzyme catalyze differ from the inorganic catalysis. Now, what is the difference, students? What is the difference between a normal catalyst and a organic catalyst? Very simple. Your inorganic catalyst like platinum, gold, and all, they can work really well at high temperature, but your organic catalyst cannot. Why? Your organic catalysts are made up of proteins, and all of you know protein undergoes what? Protein undergoes denaturation when it is too hot. A protein will undergo what? Complete inactive in the cold temperature. So proteins are very, what do I say? They want to be very safe. They can't take a lot of heat. They can't take a lot of cold also. And since enzymes are made up of proteins, enzymes will work at a very specific temperature. All the enzymes will work at a very fixed temperature. If temperature goes up and down, they'll not work properly. Similarly, all these enzymes, right, they cannot work at high temperature except one enzyme. The enzyme which I'm talking about here is none other than the thermus aquaticus enzyme Taq polymerase. Taq polymerase enzyme. Taq polymerase enzyme can work really well in the high temperature and high pressure also. Easily it can work. Taq polymerase. Okay. No. In many ways, okay, done. A thermostability is thus an important quality of such enzyme. Which one? Taq polymerase enzyme. Isolated from the thermophilic organism. Thermus aquaticus. Thermus aquaticus. Now students understand the reaction. The simple reaction is there. Right? The simple reaction is we have. Now all of you are aware that enzymes can make a reaction faster. All of you know an enzyme will make a reaction faster, but how fast? Understand the concept now. See, here we have a simple reaction. Here we have a simple reaction. CO2 plus H2O will give you H2CO3, carbonic acid. Okay. Now this reaction is happening. This reaction is happening. We are getting 200 molecules of H2CO3 in 1 hour. In 1 hour, we are getting 200 molecules of H2CO3. Okay, it is fast. But this is without catalyst. But as soon as you add a catalyst into it, example is carbonic anhydrase in the catalyst. Once you add the catalyst, what is happening? We are now getting, students, we are now getting 6 lakh molecules of H2CO3 in, here we have 200 molecules in 1 hour, but here we are getting 6 lakh molecules in 1 hour. 10 minutes, 1 minute. No students, every second we are getting 6 lakh molecules. Here it was 200 molecules in 1 hour, but here we are getting 6 lakh molecules in just 1 second. Every second we are getting 6 lakh molecules here. That is the power of enzyme. That is the beauty of enzyme. How efficiently they work, right? How efficiently they work. Okay. So it is given here also in NCERT. Let me read for you. Let me drink some water. Okay. Now see here. What is rate? Rate of reaction is nothing but how much of time it is taking for the formation of product. Rate of physical chemical process refers to the amount of product formed per unit time. Formed per unit time. So product versus time. That is the rate of reaction. If you add direction to it, it gives you velocity. If you add direction, it becomes velocity. Okay. This is what we learned. CO2 plus H2O gives you carbonic acid. 200 molecules in 1 hour. 6 lakh molecules every second. Every second, 6 lakh molecules. That is the beauty of it. A multi-step. A multi-step chemical reaction when enzymes of the steps is catalyzed by same enzyme complex or different enzymes is a metabolic pathway. For example, students, in glycolysis. In glycolysis, we have totally 10 steps. Hi, hi. Harsheen. In glycolysis, we have 10 steps, right? You have 10 steps in glycolysis. 10 steps in glycolysis. Right, all of you are aware. Now, all those 10 steps do they happen at the same time? No. It takes some time. One after another step takes place. Now, in these 10 steps, do we have the same enzyme in every step? Or every step has a different enzyme? Every step has a different enzyme here. So that is the beauty of the enzymes in the multi-step process. Okay? No. So, types of enzymes, friends. Type of enzymes. Again, very important concept called as types of enzymes. There's no simple enzyme. Very simple enzyme. They only have the amino acid group. Amino acid group. But there is friends. There is something called as conjugate enzymes. Conjugate enzymes. Now, what is this conjugate enzyme? Now, how is it different from the normal enzyme, students? Conjugate enzyme. Understand the concept. Your conjugate enzyme has two parts. A conjugate enzyme has two parts. It has a apoenzyme part and it also has a cofactor part. Now you tell me, sir, is cofactor even necessary? Can the apoenzyme function on its own? No. So in these two different particular uh, different parts of conjugate enzyme, if you have only apoenzyme, it is biologically inactive. But as soon as you add the cofactor to it, cofactor will do what? It activates the catalytic activity. Apoenzyme is protein part, cofactor is non-protein part. So once you add apoenzyme plus cofactor, when you add them together, we get the holoenzyme. We get the holoenzyme. And holoenzyme can function really well. See, if we have only apoenzyme, it cannot work. It is biologically inactive. The apoenzyme will only work when you say when it has cofactor with it. Now you tell me, you're going to class every single day with you and you're having fun with your best friend. Imagine if one day your best friend is absent, do you feel that you do not have any friends in the school? You're like, oh my god, my life is so lonely, I have no friends. Because your best friend is absent. Here also same thing happens. Here we have something called as apoenzyme. It is inactive on its own. Inactive. But when we add cofactor to it, once you add cofactor, it becomes biologically active that time. Biologically active, then it becomes. Okay. Now, next concept is how enzymes do it. Like, what is the actual reaction happening in the cell, right? What is the actual reaction happening and what is the step by process? What is the step by process? Understand. Here you have something called as a reactant one, also called a substrate one or reactant one, R1 plus R2, we get a product. This is straight up simple, very simple. But what happens is we just don't have this very simple reaction. There are always certain intermediates. For example, we have something called as substrate 1 plus substrate 2. It will give you something called as intermediate product. Intermediate product. Now, what is the intermediate product? Intermediate product is nothing but substrate plus enzyme complex. Then we'll get something called as enzyme product complex. Then we'll get the product. So what is happening here is first we getting the substrate plus substrate intermediate product. See here. See. Substrate plus enzyme, it is making enzyme substrate complex. Then what happens? Enzyme substrate complex gets converted to enzyme product complex. Then we end up getting the product. Then we end up getting the product plus enzyme. And always remember, enzyme is never consumed. Enzyme is never consumed in the entire reaction. Okay. Now see here what we learn. So what do we have? Substrate 1 plus substrate 2. In the presence of enzyme, what is happening? Substrate is forming a bond with enzyme. Enzyme substrate complex. It gets converted to enzyme product complex. Then we end up getting the product. Then we end up getting the product. Plus enzyme is coming out. Plus enzyme is removed outside. Plus enzyme is removed outside. Okay. Hello, Sneha. Diksha sir, it seems again same joke. Now, students, there's something called as activation energy now. Now, what is the meaning of activation energy? Very simple, again, understand. What is activation energy? Activation energy is nothing but it is the average energy difference between. It is the average energy difference between substrate between substrate and intermediate product. Intermediate product. For example, students, here reactant 1 has some energy, intermediate will have some energy, and product will have some energy. Now I am telling there is a clear energy difference. I am telling you there is a clear energy difference between your substrate and intermediate state. Intermediate state is also called as what? Transition state. There is an energy difference. Can you see here? First we have the reactant here, students. First we have the reactant. Here we have the intermediate and we have the product. What actually happens is the intermediate always has a higher energy. Intermediate always has a higher energy. So what is happening here is very simple. Imagine you're trying to do a high jump. You're trying to do high jump. When you're trying to do high jump, obviously if the bar is very high, you can't jump very easily. It tells you that the activation energy is very high. So what is enzyme doing? Enzyme is reducing the bar. Enzyme is reducing the activation energy. It is the average energy difference between reactant and intermediate. So it is decreasing the energy only. Who? Enzyme. So if enzyme is decreasing the energy difference, if enzyme is able to decrease the energies, see friends, reactant, this is intermediate. This is the difference, energy difference is called as what? Activation energy. Now once we add the enzyme, what happens? Once we add the enzyme, this activation energy is only decreasing. Can you see? Decreasing. It decreased till here now. Now reactants can easily become products. Easily become products. So the main function of enzyme, the main function of enzyme is what? The main function of enzyme is reducing the activation energy. Clear all of you? Tell me. That is the main function of enzymes. The main function of enzyme is to reduce the activation energy. That's how enzymes work. Now students, there is something called as endothermic reaction, exothermic reaction. What is the difference? Students, see this. In the case of your endothermic reaction, products will have high energy. Now tell me, if products have high energy, reactant will have to reach that energy level. So they will absorb more energy. They will absorb more energy and then they will reach here. So absorbing of energy is called as what? Endothermic reaction. Now if you look at the exothermic, reactants have high energy here. Reactants have high energy. Now you tell me, if reactant has to reach the energy level of product, it has to lose some energy now. So exothermic reaction. In exothermic reaction, energy is being lost. Energy is being lost in the exothermic reaction. Okay. See here, friends. Substrate S to bind at the enzyme at active site within the given cleft or pocket. See here, friends. This is the diagram. We have something called as activation energy with enzyme, very less. Without enzyme is very high. So whenever there is enzyme, activation energy decrease, substrate can become product easily. See, enzyme, substrate, enzyme plus substrate, enzyme substrate complex, enzyme product complex, and finally the enzyme and product are separate. The first substrate binds to the active site of the enzyme. Substrate is always binding, not ulta. Enzyme will never go and bind. Substrate will always come and bind. Substrate will always come and bind. Fitting into the active site. Okay. The binding of the substrate induces the enzyme to alter its shape. So once it is binding, that is, substrate is binding into the enzyme, enzyme will change its shape and tightly bind with the substrate now. Okay. Fitting more tightly to the substrate. So enzyme is changing its shape. It's like, I'll move a little here and there. and I will bind very tightly. The active side of the enzyme now is in close proximity of the substrate, breaks the chemical bond of the substrate, and the new enzyme product is formed. Okay, and finally enzyme product is released outside. The enzyme releases the product of the reaction, and the free enzyme is now again ready to bind to the, waiting for the new substrate to come and bind. Okay. Now there are multiple factors affecting the reaction fluence. There are multiple factors. For example, temperature, pH, enzyme concentration, enzyme substrate concentration, inhibitors. How each one plays a role in the enzyme activity, students. For example, I told you already, every single enzyme has a favorite temperature where it will work really well. That is called as optimum temperature. But what if the temperature becomes very high? If temperature becomes very high, all the enzymes undergo denaturation. Why? Because enzymes are proteins, and proteins undergo denaturation in high temperature. Similarly, if you make it very low temperature, what happens? If you make it very low temperature, the enzyme will become inactive. Enzymes will become completely inactive friends. Okay. Now pH also goes same thing. For example, if you look at it, pepsin has the optimum pH at around 1. Salivary amylase has a pH at 6. Alkaline phosphate has around 9. So every single enzyme has a favorite pH where it works very well. Anything more than that or anything below that, the enzyme will not work. The enzyme will not work. Every single enzyme has a very favorite or optimum pH level. Now, substrate concentration, understand. If you look at the substrate concentration, first we have enzymes here. Can you see? The first enzymes are free enzymes here. Enzyme is free. Only this enzyme is forming a bond with the substrate. Now, why is this happening? This is happening because the substrate is only very less. Substrate is less. Now, what you do is you increase the concentration of substrate in medium. Increase the concentration of substrate. Now, you add more substrate to it. If you add more substrate, obviously the chance of reaction happening is increasing now, because more substrate is binding to the enzyme now. So, can I tell in medium range, rate of reaction is directly proportional to the substrate concentration in medium. But at one point, students, at one point it becomes saturated. Why? Because all the enzymes are already covered. All the enzymes are already finished. Binding is done. Now at one point, after one point, how much ever substrate if you increase, keep on increasing 100 times also, reaction will not go fast. Okay, reaction will not go fast. After a point, after a certain point, reaction stops, or it becomes saturated. Okay, the same goes for even enzyme concentration also. The same goes for enzyme concentration also. Now listen to me very carefully. There's something called as Vmax here. Vmax. What is this Vmax, students? What is this Vmax? Vmax is nothing but maximum velocity. It is nothing but maximum velocity at which all the active sites are filled. So at what velocity all the active sites are filled? It means at what velocity enzyme reaction is the fastest. Okay, enzyme is able to work, or the product is getting converted to, substrate is getting converted to product at maximum velocity. That is called a Vmax. Why? Because at Vmax velocity all the active sites are filled. But students, understand this Vmax is very difficult to find. So what do? Instead of finding the Vmax, we do half Vmax. We do half Vmax. What is half Vmax? Half Vmax is nothing but the velocity at which, half Vmax, velocity at which half of the active sites are filled. Half of the active sites are filled. Half of the active sites are filled. That is called as half Vmax. And students, at half Vmax, half of the active site is filled. Filled by what? Filled by substrate. So at half Vmax, some amount of substrate is binding with the enzyme. Now if you want to find how much amount of substrate is binding, that is called as Km value, Michaelis constant. Okay, so half Km value is nothing but Km value is nothing but the substrate concentration. It is nothing but the substrate concentration at half Vmax. At half Vmax velocity, how much of substrate is being used? How much of substrate is being used? That is called as Km value. Km value. We learned about temperature and pH. Yes, we learned about temperature and pH. We learned here proper temperature called as optimum temperature and optimum, yes, optimum pH and optimum temperature. Below it does not work. We learned about your substrate concentration also, right? With increase in the substrate concentration, the velocity of enzymatic reaction rise at first. So initially it will increase. More substrate if you add, the rate will increase starting. The reaction ultimately reaches the maximum velocity, that is called as Vmax, which is not exceeded by any further. That is, at Vmax, how much ever substrate if you add more, reaction will not increase any further. That is called as Vmax, maximum velocity it can go. Okay. This is because enzyme molecules are fewer than the substrate molecules. And after saturation of these molecules, there are no free enzymes. So after saturation, there are no free enzymes. Obviously, the rate will not increase any further to bind to the additional substrate molecules. The activity of an enzyme is also sensitive to presence of specific chemicals to the bind to the enzymes called as inhibitors. Called as inhibitors. Inhibitor is a very beautiful concept. Again, we will learn today only. Okay. Inhibitors there. Then we have something called as type of cofactors, and the chapter is over after that, I feel. Now, students, we will take a 15 minutes break and then we'll start again. I'll go eat something and come. I'll go eat my lunch and come. We'll take a 15 minutes break and then we'll start again. Okay, what's the time now? Let's take it as 50, right? We'll start at class resume at 4:05. At 4:05, we'll start the remaining part. Could be inhibitors. We will learn something called as inhibitors. Very easy, students. Very easy inhibitors. Then I'll teach you something called as cofactor. What is organic? What is prosthetic? What is you know inorganic type of cofactors? How cofactors influence a reaction, right? And the chapter is over with that. So it is over. Nothing much is there after that. Okay. I'll see you all of you at 4:05 directly. So, hello students. Let's start with the last part of the chapter, right? We have to do now the last part of the chapter, that is basically the inhibitors. So now we learned that a particular enzyme can be inhibited if the temperature is too high. Similarly, a particular enzyme can be uh, inhibited if the temperature is too low. If the pH is too high, also enzyme can be inhibited. If the pH is also too low, that time also enzyme can be inhibited. But what if there is something called as inhibitor only? There is something called as inhibitor only, which will completely stop the function. Such a type of chemical or such as a molecule is called as inhibitor. For example, students, we have something called as inhibitor. Chemical that shuts off the enzyme activity completely. Now here we have different types. We have something called as competitive, uncompetitive, and non-competitive. Three different types of inhibitors which completely stop the enzyme activity. They completely stop the enzyme activity. For example, look here. Competitive. Competitive inhibitor is in a competition. And since it is in a competition, it will look exactly like the substrate. For example, students, see here. We have the substrate. Instead of substrate, this is substrate, right? Instead of substrate, enzyme is going on binding. Full clone. So competitive inhibitor will resemble the substrate and compete with the substrate for the active site. So imagine if this is the enzyme, and if this is the enzyme, so instead of substrate coming and binding, inhibitor is coming and binding now. So we get what? We are getting something called as inhibitor enzyme complex. Inhibitor enzyme complex. Substrate is lonely, separately it is there in the corner. Okay. Now what is uncompetitive or non-competitive? Okay. Non-competitive binds at the site other than the active site. See, this is the active site. It will bind somewhere in the back. Okay. And when it binds somewhere in the back, it will change the entire enzyme only. See here. Binding here, entire structure is changing. Can you see? Entire active site is changing. So once active site is changed, substrate cannot bind anymore. So enzyme, your inhibitor is binding other than the active site, changing the structure of an active site, and substrate is not able to join at all. Non-competitive. But what about uncompetitive friends? Uncompetitive is very simple. It will wait. It cannot join at first. It cannot join. It will wait for the enzyme substrate to happen. It will wait for the enzyme substrate to happen. Then it will come and join. See here. It will join first. It cannot join. Inhibitor, this will join. Then it will join and push it out. Then you push it out. That is called as uncompetitive. That is called as uncompetitive. It will wait. It will wait for the enzyme substrate complex to form first. Okay. Now, students. Now we will understand something called as cofactor. Cofactor is basically the non-protein part, all of you know. Cofactor is basically the non-protein part. See, apoenzyme plus cofactor is holoenzyme. The only way apoenzyme can work really well if it has cofactor. And cofactor are mainly of three types. Okay, mainly of three types. For example, students, let's read what is given here only. Mainly of three types. Okay, one is called as prosthetic group, coenzyme, and metal ion. Out of these three, this one is organic, coenzyme is organic, prosthetic group is also organic in nature. It is also organic in nature. The only inorganic one is metal ion. It is inorganic in nature. It is inorganic in nature. Okay. Now see what is happening in prosthetic. One by one example, very important. Last part of the chapter. Right. Prosthetic group. First of all, it is organic in nature. Point number one. Organic. It is very tightly bound to the substrate. It is very tight, not a substrate, tightly bound to the apoenzyme. Cofactor is bound very tightly to the apoenzyme. Okay. Example, third point, that is peroxidase and catalase. Peroxidase and catalase, which catalyze the breakdown of your hydrogen peroxide. So H2O2 is broken down into H2 plus O2. The enzyme here is what? The enzyme involved here is peroxidase. Peroxidase is the enzyme. But this peroxidase enzyme cannot function. This peroxidase enzyme cannot function unless there is the cofactor. And the cofactor or the prosthetic group here is the heme group. Heme. So unless heme is there, peroxidase cannot work really well. Okay? So for the peroxidase to work really well, we need the heme group. Okay. Heme is the prosthetic group. It's a part of the active site. That is the first one. Now, second one you see called as coenzyme. Okay, coenzyme. Coenzyme is also first of all organic in nature. Now, prosthetic was very tightly bound. Your coenzyme is very loosely bound. It is very transient. Only transient, very loosely bound. Example again is very important. Example. The essential chemical components of many coenzymes and are vitamins. Example, coenzyme nicotinamide diadenine dinucleotide, the NAD, right? And NAD plus. NAD and NAD plus are your coenzymes. Okay, they are the coenzymes. Last one is metal ion. Last one is metal ion. Again, enzyme require metal ion for activity from the coordination bond. Where is it? Example is here. See, zinc is the cofactor for the proteolytic enzyme carboxypeptidase. For this is the apo enzyme. This is the apo enzyme. If carboxypeptidase has to work really well, it needs the zinc. If there is no zinc, nothing will happen. Okay. Now, friends, there is something called as classification of enzymes also. Like how you have classification in plants, classification in animals, classification in microbes. There is also classification or nomenclature in the case of enzymes. Here we have six different types. Six different types. Okay, six different classes: oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. All of you know oxidoreductase is basically oxidation reduction is happening. Okay, enzyme which catalyzes the oxide reduction between the two substrate. So if there is a redox reaction happening, the enzyme involved here is the oxidoreductase. Now students, you might have observed in the chapter respiration plants. In the chapter respiration plants, most of the places you have an enzyme called as dehydrogenase enzyme. Even in the link reaction, we have pyruvate dehydrogenase there also, link reaction. So dehydrogenase enzyme is present in the link reaction where redox reaction is happening. Okay. Next one is called as transferase. Basically, this is Ag plus S. S will form a bond with S and G is removed out. Transferring, one is removing. Transferring and removing. Okay. Now, what is hydrolase? Hydrolase enzyme catalyze the hydrolysis of ester or ether peptide glycosidic bond. So it is breaking the bond with the help of water. Lyase. Lyase is joining. Enzyme that catalyze removal of a group from a substrate by mechanism other than the hydrolysis. So it is removing something but without water. Isomerase, all of you know isomerization. Same but different. Same chemical formula, different structural formula. Okay. Most application is there in the case of your glycolysis. Glucose 6 phosphate to fructose 6 phosphate. Isomerization, same enzyme. Last one is ligase, like DNA ligase, which helps in joining. Students. Now, students, I know this was a little bit lengthy, and I hope you have understood something. And if you have any doubts, let me know in the comment section. I will reply to each one of them and clear your doubts. And also, if you want to talk to me, you can always talk to me on the Telegram channel, Baswaraj Sir Biology, Baswaraj Sir Biology. You can talk to me there. And until then, take care, all of you.